Hi everyone. Welcome back to the third episode in our special lecture series on the biomolecules of the coronavirus. So today we're going to be talking about proteins. Proteins, I think, are probably you might be a little bit more familiar with, just thinking about training and how you have to eat protein and that sort of thing. So what is it? What is it for? Proteins are either for structure or for function inside of our cells and inside of our tissues. But at their heart, they're copolymers. They're still polymers, the same that we've talked about in class. And these are a very specific copolymer of about 20 amino acids. For us, it's 20. For different species, different things, that might be more or fewer. And in fact, some researchers now have created artificial amino acids that do functions that nature has never produced. But again, because we have, just as discussed in class, as long as there is an amine on one side of the molecule and a carboxylic acid on the other, we have the makings of an amino acid. And that side group, that R group that's hanging off that center carbon, is going to define how exactly that amino acid behaves and why we're going to inject it at that point in the longer protein chain. So the 20 common amino acids that we're really interested in, there's a bunch of different places you can go to find these in a chart. All the way from the very simple glycine, where the side group R is just a hydrogen, to alanine, where we've got it as a methyl group, to isoleucine and leucine, where we've got, let's see, 1, 2, 3, 4, so a butyl group hanging off of it. Proline, valine. So again, you can just go through and see all the different types. What's really interesting about them, some of them will have, say, ring structures on them, so aromatic rings. Others are acidic. Some are more basic. Any ones that are sulfur containing, so really cysteine is the example there. When these connect between each other, we can get a disulfide bridge, which is a very strong bond between different parts of the protein. And any one that has, say, a couple of oxygens hanging off it, like the aspartic acid or the glutamic acid here, or some hydrogens, so even the histidine, lysine, serine there, these are going to be able to participate in hydrogen bonding. So again, forming a weaker but still a strong enough bond between different parts of the protein. Why would you want to form bonds across different parts of a protein? Because at its heart, a protein's shape is going to define its function. Okay, so just a quick reminder about how these things are going to bond, how the amino acids are going to come together to form an amide functional group. In this context, we can also call it a peptide bond, because small runs of amino acids are also called polypeptides. And so when we eat these things, really what we're going to do is break down the protein, denature it, so we're going to take out its shape and just stretch it back into a long deformed chain, break that into peptides, break those peptides back into amino acids, and just use the amino acids on their own. So what's really important here is the order of amino acids in the chain. So the order of the amino acids is going to determine how those different parts of the chain are interacting, whether they've got a disulfide bond, some hydrogen bonding, some interactions between these parts of the chain. How those different regions of the longer chain interact is going to determine the overall shape of the polypeptide, which again makes sense. If you had, say, a long string with magnets just tied in at various spots, where those magnets lie is really going to depend how the whole thing comes together to make a larger shape. The shape of those parts of the polypeptides is going to determine the shape of the overall protein, whether it's just one chain or many chains. And finally, what's really most important to remember here is that the shape of the protein is going to determine its function. So a protein can lose its function if the shape changes. So that's essentially that's why we cook food. That's why say an egg white is going to go from clear or translucent to white as it cooks, is because the what was a tightly bound and well formed protein is going to lose its shape, and as it stretches out into just one longer chain, now it can cross-link and interact with other proteins to become something totally different. Okay, and because each level of structure is dependent on the lower order of structure, we organize them according to that. So we also talk about the order of amino acids in the longer protein chain as the primary structure. So just which exactly protein we see in this order is going to determine the primary structure. When we go into the secondary structure, now we're going to say how these things are going to interact. And the usual shapes that we're interested in for a secondary structure is either what we call an alpha helix. So it's this coiling helical shape, or a beta pleated sheet. So here you can see that the amino acids are actually forming a sheet with some gentle folds in it. So this would be a beta sheet or a beta pleated sheet. So this is the secondary structure of the protein. So now how those secondary structures are going to interact and shape with each other forms the tertiary structure. So this is the overall chain shape and how it is formed in 3D space. So that's the tertiary structure of the amino acids into the secondary structure of the helices or the pleated sheets, into now the tertiary structure of the protein itself. And then finally, if that single protein also interacts with other proteins to make the overall functional group, I shouldn't say functional group, that's going to get confusing, but the overall machine, let's say, that would be the quaternary structure. So the fourth order of structure of these proteins. So something like that, what's shown here might be a hemoglobin. So these would each have, say, 4 heme groups, one in each of them, making the entire protein that's going to carry the oxygen on the red blood cells throughout the body. Okay, so again, protein functions determined by shape, and according to that shape and a little bit according to the function and where you might find it and what the job it's doing, we can also classify them differently. So globular proteins are roughly spherical. They're more water soluble than some other proteins, and they can be dispersed in liquid. So it's these sorts of proteins that are giving milk its white, cloudy color. Where else do we see these types of proteins? So again, hemoglobin is a good example. Insulin, it's something that you want to be able to move quite easily in the aqueous solution that is your blood. Actin, so this is a part of the protein that's going to help your muscles move. And albumin are well, they're actually a pretty common protein, but they're the ones that give egg whites their texture when they're cooked. And actually when they're uncooked as well, but it's this when or during cooking, this tight globular shape is going to get lost, and all of the hydrogen bonds that keep this in nice order are just going to be stretched out, and what was a very tight little package becomes a long chain that can interact with other chains, and we end up with a different texture altogether for the egg white because of this network of albumin chains now. Okay, so globular proteins, they do a function usually, but you want them to be able to move around in an aqueous fluid quite freely as a colloid. Okay, fibrous proteins are a different story. So these are ones that you want to be tougher, you want to keep them in place, you don't want them to be water soluble. So now we're going to be looking at things like keratin, that it's in your hair or your fingernails, something that you want to be relatively tough. Collagen, so a lot of the connective tissue in the body is held together by this protein collagen. What do I say here? You want it to be stable in a wide range of heat or pH, so this is why it takes a long time for a tough meat that has a lot of connective tissue like collagen in it you have to stew it for a long time to break down those proteins, or else it's very tough. Same thing with pH. The interesting example here, I think, is this spideroin. So a family of proteins that are that make spider silk. So here we have a nice picture of what's going on inside a spider as it's weaving different types of webs. There's actually on the order of 7 different types of silk that spiders produce, depending on what they're trying to do. So again, you'd have different proteins, different protein structures, or different primary structures and different secondary structures that are going to make either something that's quite, let's say, quite strong, so something like a structure or the drag line that's going to support its whole body, or something that's fine, that say you want to actually fill in some of the capture point of the web. Something soft to protect its eggs, or something sticky that's going to help secure the web to different points. All of these materials have different properties, so they have to have a different protein structure. Okay, let's move away from that horrifying picture into the third major protein family. So these are ones that are embedded or on the surface of cellular membranes. These are interacting with the hydrophobic, the nonpolar parts of the lipid protein, or the lipid membrane that makes up your cell walls. So these ones are going to integrate directly into or onto the cellular membrane to allow some kind of interactions between the interior of the cell or the organelle and the exterior. So these would be things that are controlling the change in pH. So if we're controlling the flow of the protons or of the hydronium, then we're changing the pH in these different regions. We can change the concentration of potassium or sodium or calcium, and that actually works to function your nervous system, or to control your heartbeat, or to control the flexion of muscles. Is all dependent on the control of ions into or out of different regions of the cell. Excuse me. So these proteins are also used for recognition, for cell-to-cell signaling, and to control any actions inside the cell based again on some of these other properties. So I showed these at the beginning. So this first protein that would be attached onto the surface of the phospholipid bilayer, this is the spike protein of the coronavirus. So this is the one that's going to attach onto our, this beside it is a human ACE2 surface protein. So these, this set of structures, which you'll see look quite tubular. So this is another way of representing the alpha helix. These are all quite hydrophobic and are going to embed inside of our outer surface of our own cell membrane. This has a function besides just allowing the coronavirus to mess up your summer. But when the coronavirus spike protein approaches it, it has a high affinity for this pocket in the ACE2 receptor. And what happens is that it forms a bond between it, and a tight enough bond that that receptor is going to react to the presence of the virus and begin to allow it to come into the cell. So this is the virus kind of picking the lock that allows it to interact with the inner parts of our cell and to take over that machinery to do what it needs it to do. Okay, so that's kind of one of the major proteins that's on the surface of the coronavirus in that gray and red picture that's quite famous now. These are the red proteins that we're looking at. But it's not the only protein that it has on its cellular surface. Another one is a type of enzyme. So enzymes are quite special cells, or special proteins that catalyze chemical reactions very, very efficiently. They're also very specific just for the reaction that it wants to perform. So this is one of the ways that our cells can control the concentration of different molecules inside the cell or inside the body as a whole is by activating or deactivating these enzymes, so it can control how quickly or how slowly these molecules are either being produced or being degraded. So it's this is a very, very simplified version of that, where the substrates, these are the reactants when we're acting with enzymes, they can also be called substrates, come together. They fit inside an active site quite well, complexing with the enzyme itself. And the enzyme takes away any of the energy barrier, or as much as it possibly can, to make these two substrates react, and then releases the product. So it has a high affinity for the substrates, a lower affinity for the product, so that once this chemical reaction is made, the enzyme can let go of it. Just enzymes in a nutshell. These are, again, like I said, very, very effective catalysts, effectively. So the SARS coronavirus 2 that we're dealing with right now in the summer or the spring of 2020, this has a transmembrane enzyme embedded on its surface that's a hemagglutinin esterase. And so when we look at enzymes generally, it's going to have anything with an ACE on the end. So if you hear about molecules or proteins that have an ACE, so a polymerase or an esterase, what it's going to do, this one in particular, is going to break down an ester bond. So usually it's going to say right in the name what this enzyme is trying to do. So we say it has this suffix, so we know it's an enzyme. It's got the ester there in the name, so it's going to break an ester. The ester that it's going to break is one that keeps that that receptor, our cellular receptor, functioning properly. So it's going to start to break down the proteins on our surfaces in order to allow its own genetic material to enter the cell and to start causing havoc. So this is its hemagglutinin esterase, part of the system of proteins that it has on the viral surface that are working against us. So connects with the spike. This protein is now going to help to open the door between recognition, binding, opening again the cell membrane, and releasing the RNA to the viral RNA to take over our equipment and start replicating. It's unfortunately for us quite a good system. So, but it is giving us some targets and things to look for antiviral drugs and for recognition for vaccines. So that's going to be it for proteins. The next one is going to be about carbohydrates, so I look forward to talking to you then. Okay, so stay safe, and I'll see you soon.